Thank you for standing by. This is the conference operator. Welcome to the Capricor Therapeutics HOPE-2 top-line 12-month data call. As a reminder, all participants are in listen-only mode, and the conference is being recorded. After the presentation, there will be an opportunity to ask questions. To join the question queue, you may press star, then 1 on your telephone keypad. Should you need assistance during the conference call, you may signal an operator by pressing star and 0. I would now like to turn the conference over to AJ Bergman. Please go ahead.
Thank you and good morning, everyone. Before we start, I would like to state that we will be making certain forward-looking statements during today's presentation. These statements may include statements, among other things, regarding the efficacy, safety, and intended utilization of our product candidates, our future research and development plans, including anticipated conduct and timing of preclinical and clinical studies, our plans to present or report additional data, our plans regarding regulatory filings, potential regulatory developments involving our product candidates, and our possible uses of existing cash and investment resources. These forward-looking statements are based on current information, assumptions, and expectations that are subject to change, and involve a number of risks and uncertainties that may cause actual results to differ materially from those contained in the forward-looking statements. These and other risks are described in our periodic filings made with the SEC, including our quarterly and annual reports. You are cautioned not to place undue reliance on these forward-looking statements, and we disclaim any obligation to update such statements. With that, I will turn it over to Linda Marbán, CEO.
Good morning, and thank you for joining us on this call to discuss the 12-month HOPE data. Shown here is our forward-looking statement slide. Please be aware that all of our filings are updated with the SEC. I hope we find you at home this morning and doing well during these most unprecedented of times. Joining me this morning is Dr. Craig McDonald, our national PI from the University of California at Davis. Dr. McDonald really needs no introduction, as he is a world-renowned leader in the neuromuscular disease space and has presented data for Capricor at the World Muscle Society as well as other venues over the past few years. However, to remind you, Dr. McDonald is professor and chair of the Department of Physical Medicine and Rehabilitation and director of the Neuromuscular Disease Clinics at UC Davis, and of course is an internationally recognized expert in the clinical management and rehabilitation of neuromuscular diseases, which includes Duchenne muscular dystrophy, otherwise known as DMD. Now, before Dr. McDonald discusses the HOPE-2 data and its importance for patients with advanced DMD, I will take a few minutes of your time to put into context the importance of the data we have top-lined this morning. We have been working closely with the FDA for the last few years as we have been developing CAP-1002 for DMD. Along those lines, we have been granted RMAT, Regenerative Medicine Advanced Therapies designation, which is part of the 21st Century Cures Act and is similar to breakthrough designation for cell and gene therapies. We have also received orphan drug designation and qualify for a rare pediatric disease coupon if our product is approved. All of these make the path forward a bit easier and collaborative with the regulators, which is obviously critical in defining the path forward to approval. So let me remind you a little bit about CAP-1002. CAP-1002 is an allogeneic, which means off-the-shelf cell therapy product, where we think the active ingredient is the exosomes that contain nucleic acids, such as RNA, and proteins. The cells release the exosomes, which in turn track to sites of injury. From experiments that we've done over the past many years on exosomes, we know they contain multiple RNAs, primarily a few microRNAs and non-coding RNAs. These are very small sections of RNA that mediate protein expression. These are profoundly immunomodulatory and correlate with the potency of our product, CAP-1002. The cells have been in over 150 patients, and their safety profile is very good. Now, I know that this is a complicated graphic, but it's an important one because it shows the unifying mechanism of action of the cells, whether treating COVID-19 or Duchenne muscular dystrophy. Now, as you can see, the cells turn on macrophage and effector T cells, who are mediators of the humoral or blood and cellular immune response, and they modulate inflammation by controlling the release of cytokines. These are chemicals that stimulate either an inflammatory response or promote cellular repair. It is in this modulation of the immune response that we believe the cells can be effective in Duchenne muscular dystrophy, as well as in the flagrant overactivation of the immune system, which is seen in COVID-19, as well as in other indications, where inflammation is a major factor in the pathology. In fact, just yesterday, we published, along with our colleagues at Cedars-Sinai Medical Center in Los Angeles, a peer-reviewed publication in the journal called Basic Research in Cardiology, which is a peer-reviewed paper and highlights our experience using CAP-1002 in a series of critically ill patients with COVID-19 related ARDS, or acute respiratory distress syndrome. And the data from that was very promising and furthermore suggests further clinical evaluation. Now, a few years ago, some of the advocacy groups in Duchenne began speaking out about the fact that there was nothing available for the cardiomyopathy associated with DMD. And furthermore, the heart failure was found to be the #1 cause of death in patients with DMD. We were intrigued by this and embarked on a preclinical journey with the MDX mouse, a naturally occurring mouse model of Duchenne. And this preclinical work has ultimately redefined this company and our mission. Shown here is an overview of some of the preclinical work which reflects the potential of CDCs in Duchenne. What has been gratifying is the remarkable association between the preclinical data, which showed improvements and shown here in heart function, skeletal muscle function, both globally and in isolated muscle, and repair of diaphragmatic muscle damage, which compared to the data, clinical data produced in both HOPE Duchenne and our first clinical trial in DMD, and now in HOPE 2, which is by far the best clinical data we have ever seen in the clinic. In HOPE Duchenne, we focused on finding those patients that had advanced heart disease. It was a 25-patient, randomized, open-label study. Most of the patients were non-ambulant, and we dosed them one time with 75 million cells into their coronary arteries. We followed them for a year, and we studied some measures of skeletal muscle function based on the observations I just showed that were made preclinically. The data, which was published in the journal Neurology last year, showed an improvement in the scar in the heart in treated patients with a corresponding improvement in systolic wall thickening, which is a measure of how well a section of the heart is contracting. We also saw some intriguing data on the performance of the upper limb, the first version of such, which is called version 1.2, which at the time was a relatively new way of measuring skeletal muscle function in non-ambulant patients. Again, we saw what we had seen in the mice, which was an improvement in the mid or arm plus distal, which is hand and wrist, upper limb function in the treated patients, while the control patients stayed the same over the course of the year. We also identified that our best improvements in the upper limb function occurred within the first 3 months of treatment. Again, exactly what we had observed in the mice. Now, all of these observations coalesce beautifully with the discoveries that were made in the lab, that the exosomes were mediating the biology, that the cells were immunomodulatory, and that they stimulated muscle repair. We even have evidence that exosomes enter into the satellite muscle cells of skeletal muscle in the mice and stimulate repair. We also realized, and perhaps this was one of the most important discoveries, that we did not need to deliver the cells to the heart directly. We could deliver the cells by simple intravenous infusion, which is, of course, safer and easier to administer. This change in the delivery method took us back to the lab and ultimately led to our ability to do 2 things. One was to IV infuse patients, and 2 was our ability to double the dose to 150 million cells. And of course, now we were able to dose quarterly, which the data suggested that we do. So this is where my narrative ends, and Dr. McDonald will take over to share with you the HOPE-2 story. I am beyond excited for the future of CAP-1002 in DMD. We at Capricor truly believe that these cells could markedly improve the lives of those patients for which limited options exist, and we plan to work hard and effectively in bringing this therapy to approval. I will stop there and ask Craig McDonald to take over. Dr. McDonald?
Thank you, Linda. It's really a pleasure to be with you this morning and share these exciting results. Just to remind people on the call, Duchenne muscular dystrophy is a relentlessly progressive disease. The natural history has been very well characterized. And patients go through a fairly typical stage of DMD progression, from delayed and impaired acquisition of motor milestones to an ambulatory stage where they have fairly predictable loss of ambulatory skills. This CAP-1002 HOPE-2 trial has really focused on the early non-ambulatory stage where patients are actually losing overhead reach, they're losing the ability to get the hand to the mouth, and also losing hand function as well. And that was really the target population. This was really the first placebo-controlled trial in a non-ambulant Duchenne muscular dystrophy population, and the first to use the performance of upper limb measure as a primary outcome measure. So the target population, again, is the early non-ambulatory Duchenne population. This actually shows the study design. It was a phase 2 randomized double-blind placebo-controlled trial, and the objective was really to evaluate both safety and efficacy. This was the first trial, again, to use intravenous systemic administration of CAP-1002. There were 9 really excellent trial sites nationally that participated in this population. The ITT population involved 20 subjects. Mean age of the patients was 14.3 years. All these patients were on corticosteroids. 80% of the patients were actually non-ambulatory. The remainder were in the last stages of the ambulatory stage of disease. This just shows a graphic on the entry items of the performance of upper limb measure, which is actually used as the primary outcome measure here. And just to demonstrate to you what the target population of Duchenne muscular dystrophy is, the entry items on the poll actually range from a best score of 6 down to a score of 0 where you have complete loss of hand function. And the population that was a focus of our clinical trial here in Duchenne was those who had poll entry scores of 5, 4, 3, and 2. These were patients that we believed we could actually demonstrate an improvement of arm function or a stabilization of arm function in the course of a 12-month trial. Those who have complete overhead reach, those who have poll entry scores of 6, would largely be difficult to show improved arm function in the course of a 12-month trial. If we go to the next slide here, this just demonstrates really the granularity of the poll items. There are shoulder level items or tasks that are actually evaluated by the physical therapist. Middle level or elbow function items, and then distal level items as well. And the performance of upper limb is now really the choice endpoint that is being used in Duchenne muscular dystrophy to evaluate upper limb function. And this has been an evolving measure. The PUL version 2.0 had just come out at the initiation of this trial. We were fortunate enough to include both the PUL 2.0 and we evaluated function with the PUL 1.2 version. The PUL version 2.0 It has a 3-point response scale, very similar to the North Star. It's much more robust and reproducible than the version 1.2. It actually allows for compensatory strategies, so there's a focus on actual function. So it allows compensatory strategies to achieve a task. This is not allowed in version 1.2. And data that has emerged shows that it's really better able to detect a change at 12 months at all levels of ability in Duchenne muscular dystrophy. So to get into the data, first let's focus on the upper limb skeletal muscle function, measured both by the performance of upper limb version 2.0 and the version 1.2. So this is the data on the really clinically meaningful changes that were observed on the PUL 2.0. This actually combines the shoulder plus the mid plus the distal item functions. The blue bars show really very minimal decline in the CAP 1002 treated patients in comparison to the placebo patients who almost lost 4 points on their PUL 2.0 over the course of the 12 months. This represented a treatment effect of 2.4 points in the CAP 1002 treated patients versus placebo at 12 months. On the right, you can see the individual patient level data. The p-value here was 0.053. So really both a statistically significant and also a clinically meaningful treatment effect. We believe that a 1-point change on the PUL is clinically meaningful over the course of a 12-month trial. And actually, in our discussions with the FDA concurred that they were supportive that a 1-point change in the PUL 2.0 would be a clinically meaningful effect size. So we're really quite pleased to see a 2.4-point treatment effect here in the CAP 1002 treated patients. This again is the patient level data showing the treated patients versus the placebo patients. There are a number of patients that have really nice stabilization, even some improvement in arm function. The placebo patients actually had a very predictable decline over the course of 12 months. So again, this shows really the sensitivity of the performance of upper limb to actually demonstrate disease progression over the course of a 12-month period of time in Duchenne dystrophy. This actually, the next slide shows data on the mid-level PUL 1.2. This is a focus on elbow function. Going into the study, we believe that given our target population, we fully expected to see an improvement in elbow function. And again, here you see the data on the CAP 1002 patients versus the placebo-treated patients. Again, a 2.8-point difference in elbow function is measured by the PUL 1.2 in the CAP 1002 versus the placebo-treated patients. Over on the right, you can see there was one non-responder patient treated with CAP 1002, but the rest of the patients really had a very nice stabilization in function, and then the placebo patients had a very predictable decline in their elbow function over the course of the 12-month trial. This again shows the patient-level data, the CAP 1002 treated patients in terms of their elbow mid-level function. Again, you can really see the stabilization of function in virtually all the CAP 1002 treated patients except for the one non-responder. And then on the right, you can see the very predictable decline in elbow function, mid-level function measured by the PUL 1.2 over the course of the 12-month trial. Next, I'd like to focus on cardiac function. This was actually measured by MRI. And so I'm going to present data on cardiac ejection fraction measured by MRI, left ventricular end systolic volume, as well as left ventricular end diastolic volume, and then a biomarker evaluation of CKMB fraction. The MRIs were, I should mention, the MRIs were actually evaluated in a blinded fashion by a pediatric cardiologist who's really one of the leaders in cardiac MRI in Duchenne muscular dystrophy at Nationwide Children's. This individual actually read all these MRIs in a blinded fashion. The next slide actually shows the improvement and stabilization in left ventricular ejection fraction in the CAP-1002 patients. The placebo-treated patients actually had a rather significant decline in their ejection fractions over the course of the year versus the stabilization in the cardiac ejection fraction. So a downward value in this would show essentially worsening cardiac ejection fraction in the placebo-treated patients. And you can see here the stabilization of cardiac ejection fraction in the CAP-1002 treated patients. I think this is really quite exciting for us. We haven't really seen a therapeutic like this that produces a stabilization in cardiac function as measured by cardiac MRI. This is the individual patient level data. You can see interestingly, the same non-responder that did not respond to the cell-based therapy on the skeletal muscle function also showed continued decline in their cardiac MRI. The other patients showed actual stabilization, and then the placebo-treated patients all showed a very predictable decline in their left ventricular ejection fraction over the course of the trial. Also, there were measurements of left ventricular end systolic volume and left ventricular end diastolic volume that were observed. And here, dilation would actually, if you look at an indexed value, the end systolic volume on an indexed basis has actually been used as a surrogate endpoint for drug approval in adult heart failure. And here, increased values are actually indicative of worsening heart functioning, worsening cardiomyopathy. And here we actually see improvement in the CAP-1002 treated patients as measured by a mean decrease in their end systolic volume. Really, which is very exciting to see the placebo patients continue to progress. And then not only systolic function appears to be improved, but also diastolic function appears to be improved as well. As you can see on the right, there was improvement in the left ventricular end diastolic volume as measured by MRI. And again, these were really statistically significant. The end systolic volume measure was statistically significant at the p.01 level. So we're really quite excited about this data. And then finally, in looking at the biomarker data, the creatine kinase MB fraction as a proportion or fraction of the total creatine kinase observed. This is an enzyme that's known to be associated with breakdown of cardiac muscle cells. It's specific to myocardial damage. And here you can actually see an improvement in the CK-MB fraction, highly statistically significant here at the p.006 level. Increases in the CK-MB fraction really quite consistent with the deterioration seen by cardiac MRI in the placebo-treated patients and actual improvement in the CK-MB fraction in the CAP-1002 treated patients. And then again, the next slide here shows the patient-level data. Virtually all of the CAP-1002 patients showing a stabilization in this biomarker, and you can see the trend towards worsening and increasing CK-MB fractions in the placebo-treated patients. The FDA is exceedingly interested in consistency of the treatment effect over multiple endpoints, and so we also looked at measures of respiratory function, and we wanted to focus on both measures specific to inspiratory muscles, which is predominantly would be indicative of diaphragmatic improvement, the inspiratory flow reserve, versus measures of expiratory function, the peak expiratory flow reserve, or the peak expiratory flow rate, which actually is an indicator of chest wall function. And Santhera, in their studies of edevidone, have focused on the peak expiratory flow rate as a primary outcome measure in Duchenne muscular dystrophy. And in terms of the individual patient-level data, we certainly would rarely expect these measures of inspiratory flow reserve to improve over the course of 12 months. And here you can see patient-level data. A number of patients actually showed improvement in their inspiratory flow reserve, whereas the placebo-treated patients quite predictably showed a trend towards decline in their inspiratory flow reserve. And then again, the peak expiratory flow rate as a percent predicted value, you can see a number of patients here treated with the CAP-1002 product show improvement in their percent predicted peak expiratory flow rate, where again the placebo patients all showed predictable decline. I'd like to just comment on the safety data in using this cell-based product. I've probably treated more patients than anybody. We treated a number of patients at UC Davis with this product, and in terms of a total of 69 infusions were performed either in the CAP-1002 treated patients or the placebo treated patients. It was generally safe and well tolerated throughout the study, with the exception of 2 hypersensitivity reactions. There was really no permanent sequelae from this, but transient hypersensitivity reactions. In December of 2018, Capricor put a voluntary hold on dosing after 2 patients in the trials had serious adverse event in the form of hypersensitivity reactions. These were possibly linked to excipients, namely DMSO. That's the current thinking in terms of the result of this hypersensitivity reaction that was seen. There were mitigation efforts to reduce the risk of such future events. We initiated a commonly used pre-medication strategy. We actually dosed the patients with oral steroids ahead of time and then IV antihistamines to prevent or mitigate potential allergic reactions during the IV administration. And since the initiation of this pre-treatment regimen, 42 infusions of the investigational drug have been administered with only one more mild hypersensitivity reaction that just required overnight observation of the patient. So just to conclude, the HOPE-2 trial, this was really the first placebo-controlled trial showing upper limb functional improvement in non-ambulant Duchenne muscular dystrophy patients. The non-ambulatory Duchenne population represents over half of the Duchenne dystrophy patients that we follow clinically. So we're really quite excited that we actually see results that demonstrate actual improvement in upper limb function that's so important to these patients' quality of life. There were also directionally consistent improvements in strength measures of upper limb function, respiratory measures, and cardiac endpoints, both cardiac MRI and cardiac biomarker endpoints. And this is really the first ever study in Duchenne that correlates cardiac functional stabilization with a reduction in a biomarker of myocardial cell damage. And also to myself as an investigator, I really am quite pleased to see really the consistent results showing in the preclinical animal data, the phase 1/2 HOPE trial and the phase 2 HOPE 2 trial. So I think moving forward, Capricor has requested an end of phase 2 meeting with the FDA to discuss pathway to approval. They have engaged a global CMO for scale-up and manufacturing of CAP-1002. And also, we clinically believe this product is working and helping patients. So we are now expeditiously initiating an open-label extension trial of all the patients that participated in HOPE 2. Again, I think one of the advantages we have here, these patients have all continued to remain blinded. So we will actually have a off-treatment period of time where we can we have the ability to observe continued deterioration, and then we can then look at improvement in function with initiation of the investigational product in the context of an open label trial. So I think that will provide additional data demonstrating evidence of drug effectiveness as well as safety. I want to mention just the advisory board to Capricor really involved leaders in the field. Dr. Richard Finkel, Pat Furlong, Conhor actually did the cardiac MRI measures in a blinded fashion. John Jeffries, Hank Meyer, who's a pulmonologist, Eugenia Mercuri from Italy, who's really a worldwide leader in Duchenne muscular dystrophy therapeutic development, as well as Francesco Montoni, Thomas Voigt, Lee Sweeney, and Michael Taylor. So really we had really phenomenal input from leaders throughout the world. I'd also like to just finally acknowledge all the patients and families who participated in the HOPE 2 study. It was supported by a number of advocacy groups, Parent Project Muscular Dystrophy, Coalition Duchenne, Cure Duchenne, and also the trial was funded with support of the California Institute for Regenerative Medicine. And I'd like to also acknowledge the investigators throughout the U.S. who participated in the HOPE 2 trial. And with that, I'll conclude and pass the session back to you, Linda. Thank you very much.
Thank you, Craig. That was absolutely fantastic. I'd like to just say it has been an absolute pleasure working with you over the past few years on this trial. Your motivation, your commitment to patients, and your strong intellect make every time we interact a learning and growing experience for me, so thank you. Well, I'd like to close this call with a few personal comments. First of all, I'd like to thank the parents and the families of the children who participated in this trial. It's a huge lift for these people to travel to different sites. They have to take their children in wheelchairs and get on airplanes and go to different sites and go through days of testing. And they're small children, so they don't want to be in a hospital, and they don't want to have their arms poked. And so we truly appreciate their efforts and hope that we can provide the promise that we made to them, which is that we would work our hardest to get this approved. Finally, I'd like to say we had 2 positive clinical trials in the space, both showing consistency of direction with improvement in upper limb function, cardiac structure and function, and now in HOPE 2, respiratory function. We really believe that this product needs to get to these patients, and with that we have brought into our inner circle a new army of leaders, those that will help us work with the FDA to get this on an expeditious path to approval. We have already sent in a meeting request based on this data, as you'll see in the press release and as we've talked about today. Finally, I'd like to just close again with a personal comment. When I became a scientist, I dreamed that someday I would be part of developing something that could make the lives of human beings better or longer. And today I feel the fulfillment of that promise looking at this data. Let's remember that p-values really represent the chance that your data is up to chance, that you just saw this as a result of some kind of random opportunity. And what we can see here with the number of zeros before digits in many of these p-values is that it's highly unlikely that the data shown here has been as a result of chance. And so perhaps one day we can stand back and say we made a difference in the lives of those patients with Duchenne muscular dystrophy. Thank you, and please come forward with any of your questions.
We will now begin the question and answer session. To join the question queue, you may press star, then 1 on your telephone keypad. You will hear a tone acknowledging your request. If you are using a speakerphone, please pick up your handset before pressing any keys. To withdraw your question, please press star, then 2. We will pause for a moment as callers join the queue. Our first question comes from Joe Pantginis with H.C. Wainwright. Please go ahead.
Good morning, everybody, and a heartfelt congratulations for the data. Very well-designed study, very pleased to see it, and thank you for all the details, Dr. McDonald. And my first question is for you, and I think the company might be able to add on to this also. When you look at the evolution of the discussions with the FDA to this point, they've been that the FDA is amenable for the PUL 2.0 to be the primary endpoint going forward as of today. So ahead of the planned end of phase 2 meeting, and I don't want to sound too exuberant about this, so I apologize for that. How could the FDA now really ignore the cardiac data that have been generated to this point? I mean, not ignore, but basically emphasize more.
Right, Joe, thank you, and always a pleasure to hear from you. We really appreciate the support that you've given us over the past few years as we've built towards this data. Your hunch is exactly correct. Even at our FDA meeting in October of 2019, when we shared with them the results of the interim analysis, they specifically spoke about the cardiac data and the fact that this would be the first time anybody has shown any relevant improvements in cardiac function. And just to be clear, the improvement in ejection fraction is over 1 year. And if we could continue this, if we can keep these boys at a basically healthy level of ejection fraction, even if gene therapy works, which of course we are hoping for, and gets them up and out of their wheelchairs, they will need the strength of their heart to be able to sustain their greater activity. So we're hopeful that the FDA recognizes the unique and very relevant opportunity in improvement in cardiac function. And let me just say, although I don't want to be too long-winded here, the fact that we have a biomarker, a circulating biomarker in the CKMB that suggests that there is a reduction in damage to the heart actually is one of the most extraordinary findings I think I've seen in my career. Because what this basically says is that those overarching observations that we made using MRI, that the function of the heart through ejection fraction is better, that the structure of the heart through the volumes is better. We also are seeing that there's actually less damage to the heart, which is what we were associating from the reduction in scar seen in HOPE Duchenne. So I think there's every reason to believe that this therapeutic is working, and I'm strongly hopeful for its approval. Thank you.
Yeah, and if I could just make an additional comment, that in Duchenne dystrophy, even with the initiation of mechanical ventilation, we are increasingly seeing that these boys and young men are succumbing to their cardiomyopathy. That's become one of the leading causes of death. And these patients were all treated at really centers of excellence throughout the United States. All these patients were already on really optimized medical cardiac management with afterload reduction with ACE inhibitors or ARBs. They were on beta blockers, and many of them were also on eplerenone or spironolactone as well. And despite those treatments, these patients were still demonstrating deterioration in their cardiac function over the course of a year, which is certainly what we see clinically. So to actually have a therapeutic that stabilized cardiac function I think was really quite exciting to me, and should continue to be a focus as we move forward with this therapeutic.
That's helpful, thank you. And you actually did answer one of my questions, so I'm very happy to see the fact that you're going to go into open label extension study to move the placebo patients over. So having that answered already, I will ask, and I guess this is a bit hypothetical, the non-responding patient, do you have any hypotheses, or is it just a function of the severity of the disease of this patient, or anything you might be looking into?
Well, if I could just initially comment, I think that the interesting thing is that that patient, was on steroids. Despite being on steroids, seemed to have pretty significant disease progression. Again, I've not been unblinded to this, but what I think is interesting is that really the non-responder was also non-responsive in terms of the skeletal muscle endpoint, the cardiac MRI endpoint, and also the pulmonary endpoint. So it sort of the non-response was very consistent across all the measures. Linda, can you just perhaps give some thoughts about the non-responder?
Yeah, thank you, Craig, and thank you, Joe. Yeah, so the non-responder, paradoxically, although sometimes you don't ever want a non-responder, but I'll make a few comments about him. First of all, when we took him out of our analyses and reran the statistics, the p-value for the PUL mid-level 1.2 was 0.004, and for the PUL 2.0, it was 0.006. So the non-responder is clearly impacting the statistical relevance of the data. Having said all that, when I presented the non-responders' clinical history to several of our KOLs, excluding Dr. McDonald, who remains blinded, the one bit of information that I thought was very interesting is that the age of loss of ambulation prior to the use of steroids was between 8 and 10 years of age. And this young man went off his feet at 8 years of age, which is very uncommon now. Typically, it's in the mid-teens usually. And so it has been suggested that he's not responding to steroids. He also has some pre-existing respiratory disease and some other congenital issues, including some other types of muscle disease that they don't understand. And during the study, he suffered several compression fractures of his spine, so was in terrible pain throughout the course of the study. So taking together, this poor young man is really suffering tremendously, and we wish he and his family all the luck in hoping that he feels better.
I understand. It is unfortunate, and I don't know if you can bear with me for one last question. It's a bit forward-looking, obviously. As you go into the FDA now with these data, what would you say is the best case and the worst case scenarios?
Yeah, so thank you for that. The best case is full approval with a confirmatory study on the backside. What we're going to propose is a longer confirmatory study, several years, where we can really validate over the course of time the preservation of function in skeletal muscle or upper limb function, and then also the preservation of cardiac function. Very, very important indicators. We also believe, based on the data that we've seen, that pulmonary function should also track in a positive direction, all moving in the right direction. So in the best case scenario, I always say to people some version of approval because we're still working out those details with FDA. I'm very hopeful that we will be able to get this. This is not a Sarepta situation where the data does not support approval and it has to be presented that this is a great opportunity. The data supports approval, and we expect to move that very quickly in that direction. Worst case scenario is that FDA says that we need to do another confirmatory or pivotal trial, and that would be, potentially have to redo the power analyses based on the current data, but we're anticipating a 50 to 70 patient clinical trial that we would do very quickly. The interim solution is, and the one that we're banking on, is that we'll present the data from the open label extension. That will be right around the time that we would be preparing a BLA anyway, and that would support approval independent of any more data collection. So our hope is no more clinical data collection.
Got it. Thank you very much. Congratulations again, and thanks for indulging me.
Thank you, Joe. Have a wonderful day and stay well.
Our next question comes from Jason McCarthy with Maxim Group. Please go ahead.
Hi, Linda, Dr. McDonald. Congratulations again on the data. Very impressive. I just want to follow up a little bit on Joe's questions about FDA. In terms of positioning it with the FDA, I understand that the FDA, looks at every drug individually, but what are your thoughts on, with the challenges that gene therapy has faced in the last 12 to 18 months, and there's really not much out there. This is the first set of really hard data, certainly on the cardiac side. How do you think the FDA now positions CAP 1002? Because you had mentioned that we all know Exondys was approved pretty much on the unmet need, not necessarily overwhelming data. How do they look at this now?
Well, of course, I can't jump into their heads. Believe me, I want to. What I am hoping is that they realize that this serves as a very strong indicator, as you said, of preservation of cardiac function. There's nothing there. So you have unmet need finally meeting and joining with positive clinical data suggesting improvements in cardiac structure and function. This particular segment of the patient population, these later stage non-ambulant patients, are largely not eligible for gene therapy yet, and there's a lot of questions in the space, independent of whether they're eligible or not, as to whether it would work. Their muscles are so fibrose, and I'll probably let Dr. McDonald comment on that in a moment. But we strongly believe that FDA will look at the strength of the data, the strength of the opportunity, and let's just not forget that our entire world has blown up in the past few months, and traveling and participating in clinical trials is going to be virtually impossible, especially putting a boy in a wheelchair on an airplane. So we're hoping that the FDA takes all of that into consideration and lets these patients get infusions in their towns or wherever they are locally, so they don't have to travel, and then we'll show in a confirmatory study the power of the therapeutic. Dr. McDonald, I don't know if you want to comment a little further on this.
Yeah, again, this therapeutic is not targeting any specific gene deletion or mutation profile. It's agnostic to genetic profile of the patients. I think from my perspective, I think obviously gene therapy is very exciting. The trials of all the trials that are being conducted are focusing on 4 to 7-year-old patients, looking predominantly at ambulatory endpoints. I don't think the industry partners in the gene therapy space really have the dosing or the approach that you would take in trying to dose a 70 or 80 kilogram adult-sized teenager or patient with gene therapy. I don't think that's been worked out to any degree. The focus is all on much smaller children that are 4 to 7 years of age. And in addition, I think obviously the proportion of patients even in the 4 to 7-year-old age range that have positive antibodies precluding them from participating in gene therapy is at least 15%. And my guess is in the non-ambulatory population, we're probably going to even see greater proportions of patients being antibody positive. So I think a therapeutic like this, I think definitely will have a place in Duchenne dystrophy. And then keeping in mind the CK values that we've seen in the publicly discussed patients treated with gene therapy, they have very impressive decrease in CK values, but those CK values still didn't go to normal values. They go from 20 to 35,000 down to 2, 3, or 4,000. So there's still an active inflammatory component even after there's been some partial restoration of dystrophin. So I think even a modulatory treatment like this could still potentially have a place even in patients who perhaps in the distant past received gene therapy. So those are my thoughts.
So Linda, then how do you position the company now? Do you look for a partnership, or is the data overwhelming enough, that you try to move forward, on your own, or is this all dependent on the outcome with FDA?
Yeah, Jason, thank you for that. It really is dependent upon the outcome with FDA. So we've been very aggressive behind the scenes in assembling the appropriate, regulatory consultants, teeing up commercial scale manufacturing, getting, the thought leaders, including Dr. McDonald and his colleague Eric Henriksen, who is an expert in understanding quality of life measures and its relationship in Duchenne, all lined up to go to FDA and present our best case. We are also, of course, interested in partnerships at this time, whether it be for future clinical work, which we're not expecting to have to do, or for taking this forward through commercialization. But I think ultimately our current path will be defined by the response from FDA. We're hoping that FDA is supportive of the filing, because it really is to the benefit of the patients. This is not just about Capricor. In fact, it's not about Capricor at all. My wish is for the boys and for their families, because if I had a child with Duchenne muscular dystrophy, I would want them to get this therapeutic.
So last quick question for Dr. McDonald. So if you're a non-ambulatory now, a little bit older boys, young men, would you be looking to treat earlier, younger kids, younger boys, 4 to 7 or somewhere thereabouts, or do you need another trial to do that?
Well, I think obviously there's a lot more therapeutics that are under investigation in the ambulant population. So I think that the trial design would be a little bit more complicated, I think, in an ambulatory population. Obviously, I think if you're preserving skeletal muscle function, this should have an impact in ambulant as well. In my experience, I think the FDA has actually been pretty reasonable at providing broad-based labeling in the rare disease space, as long as there's sufficient safety data that supports usage in a younger population. So I do think since there have been 20% of the patients in this trial were in the late ambulatory phase, so there have been some ambulatory patients that have been treated, but my guess is the FDA would probably want to see a little bit more safety data to extend the label into younger patients.
Great. Thank you for taking the questions, and again, congratulations on the impressive data.
Thank you, Jason. Have a wonderful day and stay well.
You as well.
Our next question comes from Mark Swain with BioPub. Please go ahead.
Hi, Linda. Congratulations. Nothing seems like success. My question was already answered, and I didn't know how to withdraw it, but thank you.
You're welcome, Mark. Thank you.
Our next question comes from Dan Lin with Lin SS Management. Please go ahead.
Hi, Linda, AJ. I want to congratulate for this wonderful result. So as you said, not only for Capricor, also for the patient. I'm very pleased to see today's news release and the publication of the results. My question also mostly got answered around your path forward, but has been answered. But I just want to congratulate you for the excellent results. I'm so happy for it.
Thank you so much. We look forward to working with you and continuing to deliver on the promise for the boys with Duchenne.
Once again, if you have a question, please press star, then 1. This concludes the question and answer session. I would like to turn the conference back over to Linda Marbán for any closing remarks.
Thank you for joining us today. Thank you again, Dr. McDonald. Thanks, patients and families. Please stay well during these most unprecedented of times, and we look forward to providing updates on this program as they become available. Stay tuned, and thank you.
This concludes today's conference call. You may disconnect your lines. Thank you for participating and have a pleasant day.